or posterity. Okay. If you guys have any questions about this, please go ahead and send me a chat, or you can raise your hand if you'd like, and ask any questions. So the microbe papers, again, this is about the microbe, the one that you put on the list. I believe I've heard from almost everyone. I will send you another poking email if I don't have your microbe on there. You do not have to commit right now to that microbe if you don't want to. You have a little bit of time to change your mind, especially when you start doing some research on this. And if you're like, hey, this is not really what I thought it was, or I can't find a lot of information. I think most of you guys, sorry, I was going to mute everybody. I think most of you guys picked pretty fairly well-known organisms or viruses, so you should be able to find enough information about each one of them. How long does the paper need to be? It needs to be as long as answering all of these things. So I'll go through each one of the sections. We can kind of talk about how long each one is going to be. Depending on how many figures and things you pull off the internet, I'm going to say on the small end, it's probably going to be about 10 pages. On the large end, maybe 25 to 30. It is double spaced, so it just really depends on how much information you need to answer these questions pretty fully. So let's talk about the paper first, and then we'll talk about the presentation, because really those go hand in hand. The presentation is much shorter than the paper is, so it's just presenting information you found for your paper, but in a little bit more concise fashion. So let me go through each one of the settings on, or sorry, each one of the sections on there. Okay. So this is about your specific microbe. So this is nothing to do with the unknown project. This is the microbe you're going to pick on that lovely list I have posted. Okay. The point breakdown is next to each one of those sections, so you can kind of see how, if there's more points associated, that's where there should be the bulk of the meat of your paper, if that makes sense. So the introduction is just a brief discussion about your microbe. This should include any of that taxonomy information that I talked about kind of last time about how, you know, what class it's in. This will be different depending if you picked a bacteria, a fungal, eukaryotic organism, or if you picked a virus. Because virus particles are classified a little bit differently than bacteria and eukaryotes, you'll have different type of information than if you were picking a bacteria or fungal infection. So just keep that in mind. It's not going to be standardized for everybody because all the organisms are classified a little bit different. You should have a little bit about the discovery of the organism or virus, depending on what microbe you picked. And why you, particularly you and your partner, have some interest in this microbe. This could be something like, hey, I had hand, foot, and mouth when I was a little kid and I really remember it and it made an impact on me, or someone in your family had that. Just why you found it interesting. It could be that you just find that organism interesting for whatever reason. It would be nice to have some pictures in this section of if it's a bacteria, microscope slides of this, virus, electron micrographs of it, something like that. Did somebody pick Yersinia pestis, right? There's a lovely picture of the person that discovered Yersinia pestis. When you find the picture of that person, it is remarkably similar to a faculty member at FPU. You'll find that interesting. You might want to put that picture in there because it's just really funny how much that person looks like a certain Dr. Thompson. It's really kind of creepy. Anyway, so if you had pictures of the person that discovered it, identified it, you're welcome to put that in there too. Anytime you're taking a picture off the internet, make sure that you have the appropriate citation for that because you did not take that picture yourself. Okay, so that's the introduction section. Then we're going to move on to the physiology and structure. This is where, if you have a bacteria, we talk about it's gram-positive, it's coagulase negative, or what are the identification or important things about this microbe. Viruses, this would be stuff like, is it double-stranded RNA virus? Is it a single-stranded RNA virus? Is it a retrovirus? Those kind of things. What are the physiological characteristics about it? Is it an enveloped virus versus a non-enveloped virus? If there's tables or something that gives lots of identification of your organism, that might be a good thing to put into that section. So if you have a bacteria, again, it'll be a little bit different than you have a virus. Obviously, the growth requirements of a virus, because it's an intercellular pathogen, is not going to be the same as if you had a bacterial pathogen. So keep that in mind. The next section, this should be a nice bulky section. It is worth 25 points. We're talking about any virulence factors, so any factors specific to that organism that help contribute to disease. I think everybody picked an organism that causes a disease, so we'll be focusing on what diseases it causes. If it has a capsule, if it has like a toxin it secretes, something like that. Maybe a little bit about the virus, any sort of specific virus proteins that it has. How does it cause disease? Especially with the viruses, you might see terms like cytokine storm, or something to do with how the immune system is reacting to it. So it's not necessarily the organism, but it's how our immune system reacts to it. So you might include something like that, depending on your type of organism that you picked. Then the next section is going to be epidemiology. This is where the CDC's website and the World Health Organization website is going to be your best friends. They have most of the significant data about the CDC. That's their job, to do surveillance of who is getting what infection, if there's an outbreak of that infection, how many people are getting it, what is their demographics, like is it mostly women, is it mostly men, age group, those kind of things. There's lots of wonderful tables and graphs on the CDC website that you can pull out of there. Try to get the most up-to-date information. Sometimes that's this year won't be available. I'm going to guess the CDC is a little bit busy this year with a particular virus, so they may not have had all of their surveillance data up for this year yet, but or even for 2019. So just get the most recent data that you can get. For organisms that are prevalent, not just in the US, but in other parts of the world, like tuberculosis, things like that, the World Health Organization, the WHO, will have a lot of those statistics for outside of the US. Yeah, you can pull graphs, charts, any sort of information from CDC. I would start with the CDC and the World Health Organization. I'm guessing you can find most of the information there. If you can't, there might be a society that, like American Society for Microbiology, that might have some of those statistics if they're not on there. Yeah, so CDC does not always track every single infection. So other websites like the American Society for Microbiology, there's like Infectious Disease Society of America. They also track some of that information. So those are some other websites that you might want to look at if CDC doesn't specifically track the infection. It just kind of depends on what that infection is. And also if it's a local infection, we're going to talk about a specific infection that's called valley fever today, and the county health offices here in Fresno and the other county public health, they track that very, very closely because it affects this area more, hence why it's called valley fever, and different areas that it affects more than just the whole US. So they'd be more focused on it because it is here in this area. So just kind of really depends on what organism that you're looking at. Okay. Clinical diseases. Okay, so this is things like what does it manifest? Is it a big giant pustule on your skin, or a urinary tract infection? What kind of clinical diseases does it cause? Um, is it found in other like other hosts besides the humans? Like if it's rabies virus, talking about maybe surveillance in dogs, cats, things like that. The symptoms at each stage of infection. Most infections will have clinical progression where the symptoms may vary across the whole clinical progression. So at the beginning, you have one system or one symptom, and then it progresses to something else. Things like syphilis have a very, very complicated life cycle where there's very distinct stages along the way of the infection. Okay, so I put at the end of this, include any pictures and diagrams that might be helpful. Please, 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 anybody who has an STD, no pictures of genitalia for me here. Thanks. You can use drawings, but we're good without any of the actual detailed pictures. Okay, that'd be great. Thanks. Okay. Diagnostic, treatment, prevention, and control. This is where we're talking about how do we prevent these diseases? Is there a vaccine? Is there like a surveillance mechanism with food poisoning? How do we control food poisoning? How do we prevent that? What kind of treatments are available? Is there an antibiotic? Is there an antifungal, an antiviral? Some of you, there might not be a treatment available depending on the disease that you have. So what is the current protocol for if you get that disease? Is there control, like for example, for rabies? The control procedures are not necessarily in humans. They're controlling the animals to make sure that it does not go to humans. So this will be very specific to whatever organism, any pathogen that you picked. Then nice little conclusion section, just reiterating those points from the rest of your paper. What are those main points about it? What's kind of the summary? If it's something that hasn't been controlled or treated yet, what is maybe some areas of research that they're looking into? Like for influenza, right now we're looking for the universal flu vaccine. That's kind of the big idea. Okay, reference page is going to be in CSC format. Your references will not all necessarily be primary source research articles. They can be, and probably a couple of them should be, especially the review articles. That's kind of where your guys are going to be looking for a lot of this information. CDC website, some of the bigger diseases will have their own kind of association with that disease. That you could look at those websites as well. So these won't always be primary research articles. So keep that in mind when you are looking at these. The government websites are going to be hopefully the most accurate, as well as those society websites, American Society for Microbiology, things like that will be probably your most accurate. If you have a concern about one of the references you found, feel free to email me that reference and I can take a look at it to see kind of how valid it is. And if you're having trouble finding any information, just send me an email and I can send you some helpful direction if you're having trouble finding any of this information. Okay, so that is the paper. And then I'm going to go ahead and give you the presentation guidelines, which are fairly similar, but it's going to be just, oops, a little bit more compact form presentation. Okay. All right, before I give you these though, I want to ask my little poll question. So I have kind of consulted with other faculty that are doing presentations, and would you guys prefer to do it like this, in this lovely format via Zoom live, or would you rather record it beforehand and just post it to Moodle so everybody could watch at their leisure? You don't have to commit now, I just want to kind of get a general idea. Would you prefer to do it live, or would you like to do it recorded and posted? Can anybody vote? No voting? It's not working? Let's try again. Relaunch poll. Continue. No? Ah, yay, okay. Okay, well, that's clear. All right. So nobody likes doing this, huh? Oh, we got one person. Anybody else want to vote? Got 3 more left if you want to vote. This is like ripping off the Band-Aid though. Once you're done, you don't you don't have to re-record it like 10 times. But. Anybody else want to vote? You don't have to if you don't want to. 3, 2, 1. Okay, I'll go ahead and end it. We've got a clear winner here. Okay, so I will work on finding a really good format for you guys to record and post them, because that seems to be the clear winner of what people would prefer. Okay, so let's go back into this. All right, let me share this. Whoops. Okay, share. All right. Okay, so you, again, don't have to be in groups of 2, but if you'd like to, you can. Same thing with the paper. If you are in a group of 2, I only need 1 paper. You do not have to submit 2 different ones. You guys get to work together on that. The presentations are going to be 20 minutes. Because everything is going to be recorded, I'll have to think a little bit more about doing this, but probably the best is to do some sort of PowerPoint where you can either do a voiceover if you want to on the PowerPoint, and I can give you information on how to do that, or you can record. So Zoom has a feature where you don't have to do it live. You can record yourself kind of like I'm doing now, but you record it, you can edit it, and then you can post it. So that will be an option as well. But making a PowerPoint is probably going to be the best. So you can have the pictures and everything that you need there available. Okay. I'll give you guys a couple of options. Not everybody needs to pick the same thing of how you want to prepare this. As long as you get a video to me, that's really all that matters. So this is going to have pretty much the same information that you put in your paper. Just you'll need to pare it down to kind of the highlights of the paper. And again, depending on your microbe, there's going to be more information for some microbes. You know, Yersinia pestis we've known about for a very, very long time, and people have been studying it for a long time. So there's going to be a lot of information. You could probably spend the whole 20 minutes just talking about one of the virulence factors in there. So for those of you who have a lot of information, it's really paring it down to about 20 minutes of information, how to present. So again, what is the description of your organism? Kind of give us the Cliff Notes version of it. Who discovered it? How is it found? Where were the first infections? Kind of the growth requirements. The vector, for those of you who have viruses, we can, or some of the eukaryotic pathogens, they'll be like the vector will be mosquitoes or something like that. So you can talk about that. Who's the host? Is it naturally in us? Is it naturally in other organisms like influenza that's found in swine and birds and things like that? Talking about some of the virulence factors, again, the highlights depending on what type of organism you have, the disease, the symptoms, and how we treat it. Where is it most prevalent? Is it here in the US? Is it outside the US? Is it here in the Valley? Is it California? Things like that. If there's other types of cool information that you found along the way, you can share those as well. So the general idea is that in a 20-minute presentation, you'd have about 20 slides or so. So if you are doing this with a partner, you're trying to evenly divide up those slides. You can do that however you like. One person can do the first half, the other person can do the second half. You can go slide by slide and pick which ones you want to present. It really doesn't matter. You just want to make sure that you guys are evenly participating in that presentation, okay? I'll send some information about how the best way, if you guys want to do it via Zoom, if you want to use other programs to get the videos sent to me, we'll figure that out. Okay. I'm still waiting to hear back from a couple of people about this. Now that I know that you guys all want to record, I will, because I was going to do the Zoom sessions in the schedule like we had on the syllabus. So now that I know you guys all want to record, I will figure out a due date and I will post that for you guys and redo the schedule and have that posted later today. You don't have to keep your presentation in this exact order. As long as you present all this information, you can make it flow however you think flows best. Usually people start with the history and then move on to from there, but you can present however you like. Okay. All right. So with that, oh wait, one more question. Yes, you absolutely can choose a parasite. We're going to talk about some parasites. You can choose a parasite. Giardia is fun. Makes you burp up sulfur. Yeah, it's kind of fun. Oh, speaking of Giardia, oh, somebody's trying to call me. Speaking of Giardia, I posted the link to the YouTube video for chapter 16. You guys can watch that at any point. Again, once we figure out if we're coming back, I think I'm supposed to find out next week what the plan is. I know that a lot of schools in the Bay Area have officially closed until May 1st. If that is going to be kind of the stand of most of the state, I'm going to guess that means we're not going to come back. And all of this is going to be online for the rest of the semester. And then we'll have to plan for a final that's not face-to-face. So hopefully we'll know next week. That's kind of, we're having a big faculty meeting next week with the provost, and hopefully she'll let us know what's going on. Okay, so let me move to the presentation. Get that up there. Okay. All right, so this, before we start in on officially chapter 15, which is the fungal infections, I was just gonna review a little bit about eukaryotes. I know we haven't really talked a lot about eukaryotes in this class. Chapter 15 and chapter 16 are the two chapters focused on eukaryotic organisms. A lot of this is going to be a review for you guys. You've seen all of this stuff before. You know what's in a eukaryotic cell. You probably have seen that since high school. But we're going to talk a little bit about the differences between eukaryotes and prokaryotes before we get into the fungal pathogens and then the other eukaryotes that are in chapter 16. So who are the eukaryotes? And probably something that you may have not thought about when you're thinking about your microbes for your presentation is Helimys, which number 4 in my list here, which are little worm parasites. So if any of you guys, you think of heartworms, things like that, want to change to a nice wiggly worm parasite, that's always fun. My very first job at UCLA, I worked for a parasitologist, and he made me scan in a bunch of pictures from one of the textbooks of various parasites coming out of various parts of people's bodies. It made me never want to go to Africa ever in my entire life. But some people find those very interesting. So if you would like to do one of those type of parasites, you're welcome to. Okay, so the fungal, fungus, algae, protozoa, and the Helimys fall into our categories of microbes that are eukaryotes. And some lovely pictures of these guys. So the groups from your textbook, this is how they're grouping them, is the fungi. That includes our yeast and molds that we're going to focus on later today. The protozoa and the algae, which is chapter 16. So the fungal, those are the guys like we saw in the lab when you guys took your Sabaros agar plates and you had all these lovely fuzzy things on those plates. Those are normally your molds, and we'll talk about the difference between molds and yeast today. Then we've got our protozoa, things like amoeba, giardia, again, the one that makes you burp up sulfur. It's kind of fun stuff you can get from contaminated water up in the mountains. Plasmodia, which is causes malaria. And then our algae, these are our guys that are going to be our photosynthetic eukaryotes. When we looked at the pond water in the lab, probably lots of those little algae guys in there. Okay, so what does a eukaryotic cell have? We've been talking about prokaryotes mostly, but eukaryotes are nice complex membrane-bound organelle structures. So we have internal structures like the nucleus, and our organelles we'll talk about today: endoplasmic reticulum, Golgi, mitochondria, chloroplast, things that we've all seen before. Our eukaryotic microbes also have structures on the outside, so they also have flagella. As we'll see, they're a little bit different than what we see in prokaryotes. They also have things like glycocalyx and slime layers. They do have cell walls, and we'll talk about the difference in the cell wall. They have a cell membrane, a cytoplasmic membrane, and then all these lovely internal organs that are not found in the prokaryotes. This is just a generalized idea. This is supposed to be like a plant cell stuck together with an animal cell. It's like this crazy chimera, I guess. I don't know what the book was trying to do. I think this is from the Campbell book. But anyway, on this end is supposed to be a plant cell. And then on this down here on the bottom is supposed to be like an animal cell. But it gives you the idea that inside the eukaryotic cell, we have all these lovely organelles. We can have a flagella down here, possibly. We have our nucleus, smooth and rough ER, Golgi, mitochondria. Got a couple of chloroplasts up here on top. We're going to start with the outside of the eukaryote before we move to the stuff on the inside. So some eukaryotic microbes have flagella. They are found in protozoa, algae. Again, when you guys looked at that pond water, you may have seen little swimming eukaryotes in there. Very few of the fungal cells will have flagella, and none of the ones we will talk about today will have any sort of structures for movement. Very different from the structure of the prokaryotic flagella. Remember that one kind of turned in a circle like this. We'll see the eukaryotic one is a little bit different. So the prokaryotic one had the basal body and then the filament that came out from it like this. Very different structure of the eukaryotes. So it undulates, meaning that it kind of like has this whip-like structure as opposed to turning in a circle. And instead of having flagellin, the eukaryotes have microtubules. Cilia are only found in protozoa and animal cells, and those are the ones that kind of move like an oar. Show a picture of it in a second. Very similar architecture to the flagella, but it's nice, it's small. The flagella is much longer, where the cilia are much, much shorter. So here's a lovely picture of a flagellum. This is a eukaryote, and here are all these cilia, so they look like the little hair-like projections off of the cell. So inside the flagella, if we were to like chop it, inside we would find the microtubules. So we've got the microtubules, doublet of microtubules around the outside with those central microtubules right there in the middle. And that they move in this kind of whip-like movement, so that propelling them forward as opposed to the bacterial that kind of turns in a circle, pushing the bacteria forward. So the glycocalyx is composed of polysaccharides. It's located on the outside of the cell, similar to what we saw in the prokaryotes. This is important in adhesion, so that in adhering some of these eukaryotic cells to the environment. Could also serve as receptors for communication between cells and help it react to changes in the environment. Cell walls are found in fungi and algae, and they are composed of polysaccharide fibers, and those include chitin or cellulose, so large polymers of sugar. So not the same structure as the peptidoglycan that we find in prokaryotes. Cell walls of algae can be made of things like pectin, mannans, which are mannose sugars, polymers of mannose sugars. Some of them may contain silicon dioxide, calcium carbonate. give them a little bit more rigidity in the structure. Cytoplasmic membrane found in eukaryotes, it also includes a phospholipid bilayer. It will also have proteins, but it will have sterols to help form the rigid structure of there. Same idea as a prokaryotic cell membrane, keeping the inside in, keeping the outside out. It's got the phospholipid bilayer with the inside being the hydrophobic and the heads being the hydrophilic. Okay, so that's the outside of the eukaryotic cell. On the inside, it's going to be a much more complicated structure than what we've seen in prokaryotes because we have these membrane-bound organelles. So membrane-bound organelles, meaning that these organelles also have a phospholipid bilayer on the inside of the cell, where in the prokaryotic cell, we won't have those membrane-bound organelles. The nucleus, this is where our chromosomes are located. So in the eukaryotes, we have a membrane-bound nucleus. The membrane of the nucleus is called the nuclear membrane. It's composed of phospholipid bilayer, and it has little holes in it we call nuclear pores. The main mass of the nucleus is called the nucleolus, and it's where the synthesis of rRNA, so ribosomal RNA, and then it will transport through the nuclear pore where it's going to be finally assembled outside of the nucleus. So the ribosomes, and we'll talk about where the ribosomes are located, are not inside the nucleus of the eukaryotic cell. Within the chromosome is super condensed, having structure that is not found in the prokaryotic cells. So the DNA is bound by histones. It's compacted down into a very compact structure called chromatin. A picture of that on the next slide. And we can see those structures of the chromosomes when the cell is dividing under mitosis. Here's our nucleus. Here's that nucleolus, that big giant structure in the middle. Here are our nuclear pores. And here's the structure of the DNA. So the double helix is wound down into histones. Here, this is like zooming in on this. The histones are getting down in this very complex structure, all the way down to the condensed chromosome we call chromatids, and they are only visible when the cell is going to divide. And here is just a generalized schematic of division, mitosis. So we're making 2 daughter cells, and please don't memorize the stages of these. Just to remind you, this is how eukaryotic cells divide. It's much more complicated than what we're seeing in prokaryotes that just do binary fission. We've got a whole very complex process of dividing a cell from 1 cell into 2. Okay, so because our eukaryotic cell is so complicated, right? We've got all these membrane-bound structures in there. In order to transport molecules from one section to the cell to the other, we have to have a very complex highway system in the inside of the cell to get from one location to the other, especially when we're talking about synthesizing proteins, right? Because the DNA is inside the nucleus. After we transcribe the DNA and then we have to translate it, that translation occurs at a different location, and then that protein needs to be modified and then packaged and sent to wherever it's needed in the cell. So in order to get through the different structures of the cell to the location that it needs to go, it goes through this highway system where inside the nucleus, the DNA is stuck. The DNA does not leave the nucleus. It's transcribed into RNA. Then that RNA can leave the nucleus. It moves to the ER, where it is translated by the ribosome. The protein is made, and then it's packaged to wherever it needs to go by the Golgi. So the endoplasmic reticulum is divided into 2 sections, and it was originally divided into these 2 sections based on how it looked. There was one section of the endoplasmic reticulum that looked like it had little bumps on it, and we called it the rough ER. And then there was the section of the ER that did not have little bumps that we called the smooth ER. So it was classified into those 2 categories before we even knew what was the difference between the 2 parts of those. Well, we now know that the rough endoplasmic reticulum is covered with ribosomes, and those little bumps on there are ribosomes. So the rough ER is important for synthesis and transport of proteins. So the ribosomal and the RNA will come from the nucleus. It will dock into the rough ER, and it will come in contact with the ribosome. Where the ribosomes in the rough ER will then translate it as protein. And then that protein will be able to be transported to wherever it needs to go into the rest of the cells. Proteins can be processed and modified as well if it needs to add another cofactor on there. If it needs to add a different modification, it can be modified before it's moved to the other part of the cell. So rough ER important for proteins. Again, the rough portion means that it's covered in ribosomes. Ribosomes are important for making proteins. So the rough ER's main function is in protein synthesis. Whereas the smooth ER is more important for the synthesis, storage, and transport of lipids. So making lipids that are important for the different membranes of the cell. It also is used for detoxification of different metabolic waste products from the process of metabolism. Okay, so here's our nucleus here, big purple guy. We got our little nuclear pores. So the nuclear pore, that's where the RNA is going to come out. Then we've got our rough ER, and all those little dots right here, those little kind of reddish dots are going to be our ribosomes and the sites of translation. Then we've got the smooth ER out here, the kind of lighter blue, again for our lipid synthesis and transport. All right, so the Golgi apparatus, I'm going to show a picture of it first. This is the one we all call the pita bread, or the kind of sacks here. So this is the Golgi, and the Golgi is, picture, oh, where's my picture? I'm gonna go back a little bit. Where's my cell? Oh, I got all the way back here. There it is. Okay, so here's our nucleus. There's the two rough ER, and here is the Golgi, right? So past the smooth ER, rough ER, smooth ER, we've got the Golgi apparatus right after there. So the Golgi is important for transporting to the various parts of the cell. Okay, back to the Golgi, there it is, okay. So the Golgi apparatus is going to collect and package the proteins for their final destination, wherever that might be in the cell. Now that might be outside of the cell, that might be to a different organelle of the cell, it might be in the cytoplasm. So the proteins come in from the rough ER in a transport vesicle. That vesicle then is gone to the Golgi. The Golgi can modify those proteins anymore if they need a polysaccharide, if they need a lipid, if they need to be changed in any way. And then they leave the Golgi in another vesicle, and that vesicle sends them to their correct location. So you can kind of think of as the Golgi apparatus kind of like as the UPS or the Amazon of the cell, and it receives packages, it packages them up in their own packaging, and then it puts a sticker on for its final destination. Is that going to be outside the cell? Is that going to be in the membrane? Is it going to be to another organelle? It's going to sort and modify in its own way and then send it out to the rest of the locations in the cell. So here's that vesicle that's coming in from the rough ER. It's going to fuse in the Golgi, and then here's a little secretory vesicle that will package it and move it on its way to anywhere it needs to go in the cell. The mitochondria, this is where eukaryotic cells are going to do the majority of their cellular respiration. So they're going to come into the mitochondria, and its main job is going to be for the generation of ATP for the cell. So when we talked about metabolism a long time ago, we talked about glycolysis, citric acid cycle, electron transport, etc. And in the bacterial cell, that all happens on the cytoplasmic membrane. But in eukaryotes, those processes happen in the mitochondria. So this is all location. not glycolysis, but electron transport, oxidative phosphorylation is happening inside the mitochondria. And there are more than one mitochondrion in the cell. So there's multiple in there, not just one. The enzymes for the electron transport chain, so all those electron carriers involved in passing the electrons, those are going to be located in the inner portion of the mitochondria. Mitochondria have their own DNA. They have their own ribosomes, and they have all of those enzymes important for metabolism located within them. Interesting enough, and as we get into the evolution of the mitochondria, one of the unique features about mitochondria and the fact that it does have its own DNA is that the DNA of mitochondria is in a circular form. So the DNA within the mitochondria looks more like bacterial DNA. If we look at the genes, if we look at the structure, than it does eukaryotic DNA. So here's a lovely picture of a mitochondria. We've got two membranes. So the outer membrane here of the mitochondria, and then within the mitochondria, we have a second membrane, the inner membrane. So having two membranes within the mitochondria. Chloroplasts are found in only organisms that do photosynthesis. So photosynthesis, the location of photosynthesis is in the chloroplast. Just like the mitochondria, it has two membranes, and the inner membrane is composed of sacs called thylakoids, and they stick together to form almost what it looks like Oreo cookies on top of each other, the grana. And that's where the chlorophyll is going to be located for capturing of the light energy. So here is the chloroplast. We've got the outer membrane and then the inner membrane, and we got those thylakoids stacked up on top of each other. So like if I would cut a stack of Oreo cookies in half, that's kind of what we're looking at. And then here are the Oreo cookies all together down here on the bottom. All right, so where did the mitochondria and the chloroplasts, where do we think they came from? If you remember this slide from last time, here's our last common ancestor, and here is our three domains, right? We've got our bacterial domain up here, our archaea, and eukaryote. So both the bacteria and the archaea, so our red line and our orange line up here, do not have membrane-bound organelles, right? They're both prokaryotes. So somewhere along the way, the line that became the eukaryotes here in the middle picked up these unique organelles, the mitochondria and the chloroplasts. And so what we believe is that at some point during evolution, a larger eukaryotic cell engulfed a smaller prokaryotic cell and enslaved it, or in an endosymbiont, it was in symbiosis with each other. And later that formed into the mitochondria. That a second event happened with a photosynthetic cyanobacteria where it was engulfed and later became the chloroplast. So that theory of how those two organelles evolved is called the endosymbiont theory. We think that they are evolved from the prokaryotes through that process of endosymbiosis. So what is endosymbiosis? Well, endosymbiosis, I know I said enslaving, but it's not necessarily enslaving. The idea of endosymbiosis is that the larger eukaryotic cell was providing something to that prokaryote, while that prokaryote was providing something to the larger eukaryotic cell. So it may be that the larger eukaryotic cell was providing a safe environment for that prokaryote. So maybe an environment that did not have oxygen. Oxygen is a little detrimental sometimes to some of these processes, so it might have provided that environment for the prokaryote. And the prokaryote provided things like ATP for the cell in return for that safe environment. So endosymbiosis, meaning that they're living together, each benefiting from that process. Why do we think that? Well, a couple of reasons. If we compare that DNA of the mitochondria and the chloroplast, again, they both have the circular DNA, which looks more like prokaryotes. They both have the double membrane. So they think it was evolved from a cell that already had its own membrane and then picked up a membrane as it was coming inside the cell. If we look at the structure of the genes within the mitochondria and the chloroplast, they're more similar to the genes of prokaryotes. And if we look at the genes for specifically the ribosomal RNA, we can see that it's more closely related to prokaryotes than it is to eukaryotes. So that's some of the evidence that leads to this idea that the chloroplast and the mitochondria evolved from prokaryotic cells. This theory was developed by Lynn Margulis. I believe she was at Berkeley at the time. I'm trying to remember, Berkeley or Stanford. I can't remember. Anyway, this is another kind of example of that. So we think that it was two evolutionary events that led to the mitochondria and then the chloroplast. So we got the mitochondria first, and then some of the eukaryotes that later developed into the ones that can do photosynthesis. So plants and then specific algae that can do photosynthesis picked up that second endosymbiont that became the chloroplast. So two separate events. All right, so just to wrap up this idea of what eukaryotes versus prokaryotes, this is a nice little table from another textbook that gives the differences between the two. So it's a good one to review when you're looking at, hey, what's the difference between a prokaryote and a eukaryote? All right. So let's talk about specific examples of some eukaryotes, and we're going to talk about fungal cells or fungi. Okay. A lot of people don't think about fungi too much. It's not necessarily a fun one on anybody's list to learn about fungi. Any of you that's ever had a fungal infection, you know they're very hard to get rid of. They're a pain in the butt, and they tend to last for a long time and come back. So fungi are never our friends usually. They piss us off because they grow on our food in our refrigerator. We got to throw it out. It's never fun. Okay, so fungi. Fungi, the study of fungal cells is called mycology. If any of you are like, yeah, that's what I want to study when I grow up, mycology. You can go get a PhD in that. Fungal cells are what we consider chemoheterotrophs, and mostly they eat decomposing organic matter. So you can think of your bread in your refrigerator. It's not necessarily alive, right? And so it's breaking down that food for you. Some fungal cells can go in like in between your toes, like lovely athlete's foot, and eat those decomposing cells, organic matter from your skin cells. And I have a couple of gross pictures of some fungal infections as we get a little bit farther in. They can be aerobic or they could be facultative anaerobes. And facultative anaerobes like baker's yeast, Saccharomyces cerevisiae, they can do fermentation that we use to our advantage when we're making things like beer and wine. All right. Some structures of the molds. We'll start off with these little mold guys. And for those of you who on your Sabros augers, probably most of you had some mold colonies on there. So these are our guys that are nice and fuzzy. They like to spread across a plate when they're growing, or spread across your bread if you have it in a nice warm area. Okay. So down here in the purple is the spore, right? And that's our origin of our fungal cells, from our mold. That's how we get a developing mold. So when you got your Sabros auger, you left the lid open, a spore fell on there, and the spore is then going to develop into the mold. Okay. So as the spore starts developing, this little structure that's coming out of the spore that ends up in those hair-like projections are called hyphae. So those hyphae spring out from the spore, and you can see it's gotten bigger and bigger. So almost like a seed and the hyphae are like a tree that are growing up from the seed there. We group the hyphae into two kind of classes. One that have separations we call the septa here. So these are separated hyphae, the separated hyphae here. And here it looks like almost like we're going to build a snowman with little cells on top of each other. And then ones that are connected and do not have the separation on them. Okay. But these are all branching out into different structures we call the hyphae. As we talk more about the different types of spores, sometimes the hyphae at the end will have sacs full of spores, or they'll produce a spore at the end in order to spread it into the environment and make a new mold cell. Right, and you guys may have seen these underneath the microscope in the lab. This is just a picture of like a lovely mold colony here. You see how it's spread everywhere. And if we looked at it underneath the dissecting scope, you guys might have seen something like this. You can see those aerial hyphae, meaning that they're coming up into the air. And the little black guys being the spores at the end of it. Oops. Right, so those are the molds, right? So the molds are hair-like, they make the hyphae, they come from spores, they're making the mycelia coming out from the spore cell. The yeast are not filamentous, meaning they don't have those hyphae, but they still are in the group, the fungal, they're still fungal cells. They're unicellular, whereas we just saw, we could see more than one cell on those hyphae there, we can have them separated. Yeast are just single celled, and either they bud, like Saccharomyces cerevisiae that you guys probably saw in the lab with the little bud that came off, or they just divide by fission. So the bud meaning that it's unequal division. So the little bud kind of is smaller than the original cell, and that it eventually grows into the same size, where fission yeasts just divide into two cells. Then we have some that go both ways. They're called dimorphic fungi, and they can grow like a yeast at certain temperatures, and they can grow like a mold at other temperatures. We call those dimorphic. So this is an example of a dimorphic fungi. Here's the yeast-like growth, so the little circles there, and then here's the hyphae, which is mold-like growth. And this dimorphic fungus grows like this, yeast-like at a higher temperature, and mold-like at a lower temperature. All right, so what's the fungal life cycle? So fungal cells can reproduce both sexually and asexually. So asexually meaning that it's not combining its genetic information with another organism. Basically, it's just dividing into two cells. They're genetically identical. But they do reproduce both ways via the formation of spores. That spore will detach from the parent, it will move on to a new location, and then germinate to grow a new mold. So just like your plates, the mold spore fell on there, it made a new mold. Okay, we'll talk about asexual reproduction first, and then we'll talk about the different forms of sexual reproduction. So asexual spores are just formed via mitosis and regular cell division. They're formed by the hyphae of one organism. We define the spores, these asexual spores, depending on what kind of structure the spores is made into. I'm going to show you the picture on the next slide. This is just a description of the actual structures you'll see on the next slide. When you guys looked at some of these underneath the microscope, the ones that you got from wherever you opened your plates for, you may have seen some of these. These are lovely color enhanced pictures. These are a little bit more pretty than what we looked at underneath our microscope. So the candida here, the little spores are the little, it almost looks like if you guys had the dandelions and you blow those little dandelions off into the world, and it's got that tuft of all those little seeds on it. This is kind of a similar structure like that, where they're spores and they get distributed through the air or through the environment, and they all can be released to form a new mold structure. This particular one is Aspergillus. This is one of the ones that we would consider bad if we had too much of this in our house. So that is the Condia here. They're not enclosed in the sac, they're just ready to go. We've got the little spores here, these little orange guys here. Oops, wrong direction. So these are just they're fragmented sections of the hyphae. So they've got hyphae that are segmented, and one of those sections is ultimately released to become a spore. The sporangiospore is this one down here on the bottom. It kind of looks like a puffball. These are all enclosed in a sac. Then the sac is burst open and the spores are released. So these are just the different types of asexual spores that can form from different types of fungal cells. Candida albicans, this is one of the lovely dimorphic fungus that can cause yeast infections and thrush. All right, so those are our asexual spores. Again, we'll define each one of our spores based on each one of our fungals. We'll put them into one of these categories based on how they form the spore. So if we were going to identify any of the molds that you guys had growing on your plates, we would say, okay, what kind of spores does it look like underneath the microscope? Let's try to identify it based on what structure it's forming underneath the microscope. And does it look like one of these? Well, then it possibly falls into this category. And we use that as an identification method. All right, so those are asexual spores. Again, they're just formed by a portion of the cell dividing via mitosis, then that spore is released. There's no change in the genetic information. It's identical to the parent. Where sexual spores are going to be the fusion of two different cells by different organisms and form into a unique genetic individual mold. So there's 3 different stages of sexual sexual reproduction. In that we're having, instead of male and female, we have plus and minus. So we have a plus strain of the fungal cell and a minus strain coming together to form a zygote. Then that zygote is able to replicate and form a new mold. Oh, I thought I had a picture of that. I guess I don't. I do have a couple of pictures of specific examples, but the idea is a donor that's positive interacts with a recipient that is negative. They have half of the genetic information. That those two fuse into a diploid zygote. That zygote then is able to replicate and form. The key point of the sexual reproduction is that there's 2 different organisms coming together to make a genetically unique individual, whereas asexual, it's just dividing, it's spreading its own unique information and not combining with another organism. A couple of the adaptions of fungal cells that you guys experience with those Sabaros agar plates is that they tend to grow better at a lower pH, and that's why Sabaros agar tends to grow just molds or yeast, because it has that lower pH. They can metabolize very complex carbohydrates. They can grow in high sugar and high salt concentrations, which is great for making some of the fermented things that we like to use them for. So Zyomycota, we're going to talk a little bit about these guys. They can produce spores asexually and sexually. And they have, look at this lovely picture of him. So here is it has 2 different types of reproduction that it can do. So here is the asexual reproduction on this side. Sporangiospore is released. It's just released through normal mitosis. Then it also can go into sexual reproduction, where it can form with another, sorry, I was getting a message there. It can form with another cell. Here's the plus and minus, and they can come together and form another organism that is then genetically different from the original. These microsporidia are important because they are obligate intracellular parasites, meaning that they are required to live inside a host. They do not have their own mitochondria, so they're a little unique in that fact. There's no sexual reproduction observed, which means it probably occurs in the host if there's two different types of strains available. So these guys are only going to live inside a host. And this is usually true of all of the fungal infections that infect individual, is that somehow the spores get brought into the host and they're usually inhaled. And when we talk about valley fever in a little bit, we'll talk about that is usually inhaled from, if you're digging up dirt, you can inhale the spores and bring them into your body. So normally you're ingesting or inhaling some of the spores, and then they're going to develop into the hyphae inside the body, or develop into the full-fledged structure from the spores inside the body. So this is an obligate intracellular parasite. It requires going through the host for the life cycle to complete. We're gonna see how many more. I'm gonna skip a little bit of these and just go to the disease real quick. Don't worry about too much of these. They're just kind of some of the examples of different types of fungal infections. If you are going to pick one of these, you can look at these as your life cycle of the fungal cells, but I'm going to jump into the infections by fungal cells and talk about those, and then we'll go ahead and leave it for today. So the fungal diseases, mycosis is a fungal infection. Many of you have had a fungal infection before. So you may be familiar with some of these. You may have a fungal infection now for fun. All right. So most fungal infections don't tend to be deeper in the body. We'll look at a couple of the very prominent ones that you may have experienced yourself, which are like athlete's foot, which occur on the skin. But sometimes mycotic infections can go systemic and cause a system-wide disease within the body. But most of them are cutaneous or subcutaneous, meaning that they're in the skin or at the very surface layer of the skin. Things can affect like the hair shafts, your skin, your nails, fungal infection of the skin, nails that I'll show you the lovely pictures of in a slide or two. Some of them can be very superficial, usually can be removed with creams, hopefully, and not go any deeper than that. Some fungal infections can be opportunistic, and they are causing huge problems in people that have a compromised immune system. These are some medically important fungi, ringworm, athlete's foot. If any of you have had this, we're going to talk about valley fever in a second. Again, Candida albicans causes yeast infections, thrush. We normally think of yeast infections in the vagina, but thrush is kind of the sort of similar thing, but inside your mouth. Aspergillus, that's the one I mentioned, that if you have aspergillus in your house, that's not necessarily a good thing. It does cause a chronic lung infection. So the people that had mold in their house and had to remove the mold, the one we usually associate with those mold infections are aspergillus. Histoplasma and other respiratory infections, but we'll talk about valley fever in a second. All right, here's the cutaneous mycosal infections. This is tinea pedis, means this is the athlete's foot. Ringworm, you can get this in your nails and in your skin. These are lovely pictures of very, very bad infections. Some of you may have had athlete's foot. It's just annoying and it itches, and hopefully you can put a cream on it and it goes away. Sometimes it takes a long time for these. Nail infections tend to take a really long time. One of the hard things about treating specifically fungal infection is because they're eukaryotic cells, they don't have the unique features that prokaryotes have, right? So we can treat prokaryotic cells with antibiotics that just target prokaryote-like structure, so the ribosome and the prokaryotes, peptidoglycan. Well, fungal cells being eukaryotic, a lot of their structures are very similar to ours. So if we're going to take a medication that would disrupt the DNA replication of fungal cells, it's probably going to disrupt the DNA replication of our cells too. So we have to be a little bit more careful when treating fungal infections. So normal treatment for fungal infections at cutaneous level are creams. So we put a cream on there, hopefully it will get rid of it. It doesn't usually go away very quickly. It takes many, many months of reapplication to get those infections to go away. Okay, so let's talk about valley fever. So this is the first of the diseases that we'll talk about that were named after the Central Valley. We get to have two of them. Tularemia is the other one. This is the first one that was named valley fever because of the Central Valley. So Coccidioides immitis is the cause of valley fever. At 25 degrees, it forms a colony with lots of branching hyphae, so a lovely hyphaeated structure. And then at higher temperatures, it forms these block-like structures. Oh, I thought I had a picture on here somewhere. Somewhere maybe? There it is. Okay. So these are the block-like structures with the spores inside them. They're usually found in areas with farming activities. So guess what? That's like the entire Central Valley. 60% of the people that are infected with it have a primary pulmonary infection that goes unnoticed. So cough, kind of a cold-like symptoms, nothing to be write home about, just a unique cold. In 40% of people, it might get a little bit more where you're having fever, cough, chest pains, but most people fully recover. In 5 in 1,000 cases, you can have a very severe infection, including fungulomas that can form in your lungs. So these big masses of fungal cells inside your lungs that inhibit you from breathing. So most people that get valley fever, and all of you that have lived here almost all your life, have grew up in the Central Valley, or came here while you're relatively young, you probably all have had or been exposed to valley fever. Pretty much anytime you're going to dig up the dirt, you're going to be in the dirt, you're going to be exposed to this. It's just all over the dirt in the soil. Most of you probably had it when you were very young. It was a very mild infection. You didn't even really notice, and now you've got protection against it. The severe cases tend to be people that come to the Central Valley when they're older, that they grew up in an area without valley fever. And so they're exposed to it when they're old and they do not have antibodies built up to it, and it becomes a very severe disease. If you do get it and you need required treatment, they try to treat you with antifungals. Doesn't always help. Sometimes it does, but it's more maintaining the respiratory, the airway, making sure that you can breathe. Here's some example of those fungulomas. So they can grow on your face, they can grow on your legs, and they can grow in your lungs. This is just a kind of cartoonish drawing about how those come into your body. So here are those spores. You're digging in the dirt because, I don't know, that's what everybody does in their spare time. I don't know. Digging in the dirt, the spores come up, you inhale them, they germinate within your body, and then you can develop a disease from that. Okay, so this is going to be a nice little example of how you guys can talk about the history of your infections when you're talking about your microbial infection. This will give you a little bit of kind of snapshot of how you can do that. What's the history of valley fever? So the first reported of disease here in the US was a 40-year-old native of the Azores. So this man, the Azores out there in Europe, near Portugal, island off Portugal. So he did not grow up in the area. He entered into San Francisco hospital with those crazy skin lesions, those fungulomas on there. So that was the first initial report of the disease. In 1886, after immigrating to California, that person was laboring here in the Central Valley and developed a slow, enlarging sore on the neck, and then on the eyebrows, and then swelling in both of his eyelids, so all these fungulomas everywhere. He became very disfigured, lost both of his eyes, his nose, and much of his upper lip. So it was our first report of valley fever. So the person that discovered it, identified it, cultured it, decided to name it after valley fever, because this is where this guy ended up coming from. He had been working in the valley, so that we got it named after us. Lucky us. Again, there's still valley fever up and down the valley. Most of you have probably been exposed to it. Some of you that maybe moved to Fresno just to come to school, you may have experienced respiratory disease, maybe not too severe. It may have been valley fever. Usually if it doesn't get so severe where you need to be hospitalized, you would never know that you had it versus another type of virus. All right, so not just fungal cells can cause disease, they're also beneficial to us. We enjoy the benefits of having fungal and yeast cells, Saccharomyces cerevisiae probably being the best one out of those, in that we use it to make beer and wine. We use it to ferment, to make alcohol or not alcohol. We can also use it as a very nice model organism. Saccharomyces cerevisiae is one of the most well-studied organisms because we use it as a kind of a model organism of eukaryotes in the lab. We've used it to grow things like vaccines for us. A couple of other things in there we can use. A fungal can kill termites, for making cellulose, for making other biocontrol agents to kill other organisms and such. So they're not all just bad fungal infections. They can also do good things for us. Okay. Whew. Ended that a little bit. Okay, so with that, that is where I'm going to leave chapter 15. Again, chapter 16 is posted on Moodle. I will update the schedule today, and I'll send you guys an email as well as posting it on Moodle. So you guys will have that schedule. My computer's yelling at me about something. Does anybody have any additional questions? No, nothing. Okay, so everybody should have received a link to an unknown folder. I will start dropping files and pictures and information into those folders. If you did not receive a link from me, that means I didn't hear back from you, and I'm going to assume that you want to work on that project by yourself. If you want to email me real quick and you haven't done that yet, go ahead and do that. And I'll set up a folder for you. We only have 6 unknowns. Not all of them came in on time. So some of the groups might be receiving the same unknown. So you may be able to collaborate with more people than just 2 if you find out who else has your same unknown number. So there might be a couple more sharing in there. And that's really just for you guys to discuss what you think the results are. I plan to be showing you positive, negatives, and then your unknown, so you can kind of make the decision of what you think it is, positive or negative, okay? And we'll start that on Tuesday. You should get your first results on Tuesday. Raina said she grew everything, so hopefully they all grew. The first set of results, and I'm gonna give you a nice table of when all the results will come in. The first set of results, as long as Raina and I can make them successful, are gonna be a gram stain, a spore stain, an acid-fast stain, and a simple stain. We will send you pictures of the colonies on a plate, and I think she's going to do slants as well on Tuesday, so we should have pictures and videos of that posted in your folders on Tuesday. If we can, I'm hoping Raina and I can successfully gram stain. Fingers crossed. So we know what they're supposed to be, so we know what they're supposed to look like, so that helps. Questions. No, we're not meeting on Zoom. The PowerPoint, yes. The PowerPoint for 16 is just on Moodle. And then the link to the video for 16 is on there too. It's a YouTube video you guys can watch. We will meet next week, starting with immune system, the host diseases and host function next week. Okay. Any other questions? All right. I will see you guys next week. Email me if you have questions.